Hello, everyone. Today we will talk about our immune system. There are two most complicated systems in our body that we know little. One is the nervous system, and the other one is the immune system. With a lot of development of immunotherapy in recent years, immunology will have a big progress in the next decade. In this lecture, I'll give you some basic introduction of our immune system. And for next lecture, I'll focus on several immune-based therapeutic technologies. As I know, researchers with immunology background are very easy to find a job these days. So if some of you are juniors and are interested on this topic, I suggest you to take our immunology class next semester to learn more. Our immune system is a host defense system compromising many biological structures and processes that protects against the diseases. Okay, the immune system is made of organs, tissues, cells, and molecules that all work together to generate an immune response that protects us from microorganisms, removes toxins, and destroys tumor cells. So organs and tissues of the immune system are shown in this picture, okay? So among them, the bone marrow and the thymus, they are the sites where immune cells develop and become mature cells. So bone marrow and thymus are called primary lymphoid organs. All right, so and the spleen, lymph nodes, and the specialized sites in the gut and other mucosa tissues are sites where the mature lymphocytes first meet antigens and become activated. So these sites are called secondary lymphoid organs. Okay, then two circulatory systems, namely blood and the lymphatic vessels, connect these organs, united them into a functional whole. So this is the big picture. I would also like to introduce these several types of immune cells. This will help you to better understand how our immune system works. Okay, as I mentioned, bone marrow is the primary lymphoid organ and the major site for blood cell differentiation. So in our bone marrow, there are many stem cells shown. This is just a few of them. And the process of blood stem cell differentiating into the mature blood cells is called hematopoiesis. Okay, or you can say blood cell development, the same meaning. Okay, so this is a process involving many steps of differentiation. We learn the stem cells, and you should know that all the stem cells, or sometimes called the progenitor cells, shown in this inside the bone marrow, show the green background. They are multipotent cells, right? So like blood stem cells, they can differentiate into myeloid or lymphoid stem cells, and then they will further differentiate into lymphoblasts. However, these stem cells, they won't be able to differentiate into other types of tissue, such as like say muscle or the nerve cells. So indicating they are not a pluripotent, but they are multipotent. Remember the concept, okay? So the final fully differentiated white blood cells, including those neutrophils and eosinophils, basophils, or B cell, T cell, or NK cell, also is a natural killer cells. Those are immune-related cells, okay? I'll discuss their function in detail in a few minutes. There are several levels of defense against the infection in our immune system. Skin is the largest organ in human body and functions as a physical barrier to pathogens. You know, all of us have skin everywhere, right? Epithelial cells that line in internal body cavities and ducts, such as the lungs, they are coated with mucus that protects against the infection. So the enzyme lysosome, the enzyme that can break down cell wall of bacteria, is very abundant in secretions, including the tears, saliva, human milk, and mucus, and can kill invading bacteria. Okay. If the pathogen makes its way to go inside our body, okay, our body will start to fight with innate immune system first. So then they will fight with the adaptive immune system. Okay, so the innate immune system acts very quick, but non-specific, meaning that although they distinguish a pathogen from a human cell, they don't distinguish one pathogen from another. But in contrast, the adaptive immune system has very specific responses. Okay, so it recognizes particular pathogens and responds to neutralize or kill the invader. The adaptive immune system has two components shown here. One is the antibody-mediated immunity, and then the other one is called the cell-mediated immunity. We will discuss them individually, but remember, each level of these immune responses becomes increasingly aggressive, okay? So this slide is actually very important that we will maybe come back to all these concepts later on. Okay, so this slide shows the timeline and the cells involved in innate and adaptive immunity, showing the two different parts. The innate immunity is very fast and comes into action immediately or within hours of a pathogen's appearance in the body. Then epithelial cells, shown here, and phagocytes such as the neutrophils and the macrophages or dendritic cells, natural killer cells, and also complements, those are involved in the innate immunity. The adaptive immune response is specific, but much slower response. It usually acts a day after infection, and the top cells shown this blue, those are involved in antibody-mediated immunity. So they are B lymphocytes, sometimes we just directly call B cells, okay? So once activated, the B cells become another type called the plasma cells, so they are bigger, okay? The plasma cells and produce this protein called antibodies, okay? Remember I showed you an image before when we talk about the endoplasmic reticulum. It's in the lecture 2. Inside the plasma cells, the ER becomes much more abundant, and the ribosome actively producing proteins. That's the antibodies shown here, okay? On the bottom here, this is a simplified cell-mediated immune response. Basically, the APC is also, this is abbreviated for antigen-presenting cells, namely those phagocytes and dendritic cells in the innate immune system. When they engulf the pathogen and digest them into pieces, this short peptide from these pathogens will be presented to the cell surface, okay? It's actually here. This is an empty one, but this is one with the peptide. So they can be recognized by these T lymphocytes, okay? The interaction activate the T lymphocyte and then stimulate its proliferation and then become effector T cells. And then they will make more cells. We will talk about this process in detail just a little later. Now let's discuss several events happening during the stage of the innate immune response. Inflammation has an important role in innate immunity, okay? We all should have experience with inflammatory response, right? So these slides list the stages of inflammation, and I will use an example to show the process in the next slide. So say you have a splinter penetrates into the skin and bring in the bacteria, showing this yellow thing. The bacteria will bring it inside the body, okay? First, the phagocytes, such as the macrophage showing here, will go off and destroy bacteria and secrete cytokines. Okay, so this small picture showing here shows you how a macrophage cell engulf a pathogen. So cytokines here are small protein secreted by immune cells and have an effect on other cells. All right, the chemokines are just a family of very small cytokines. So they are signaling molecules for immune cells to communicate with each other and function as basic substance for inflammatory response. These cytokines stimulate mast cells. They're showing this yellow colored cells. So these mast cells, once it's stimulated, they will secrete histamine. Okay. Meanwhile, cytokines and histamine dilate these capillaries to increase the blood flow, and then allowing the neutrophils to penetrate or migrated into this infection site and engulf the bacteria. After bacteria are cleared up, the clotting factors seal off the infected area, and the phagocytes clean up debris from dead cells. So the neutrophils actually are the most abundant white blood cells in our blood. After fighting with bacteria, the dead neutrophils could form pus, which is a positive indication for bacterial infection. You may all experience that already, right? So when we see this pus, we know, oh, this is a bacterial infection. I need to use some antibiotics to treat it, right? The complement system participates in both non-specific and specific immune response. The complement system is a multi-protein cascade of proteins that enhance the immune system to clear microbes and damaged cells. All right, so the complement system consists of about 20 to 30 different proteins synthesized in the liver and then secreted into the blood as inactive precursor proteins. When stimulated by trigger, protease in the system cleave specific proteins to release cytokines and initiate an amplifying cascade of further cleavages. At the end, the cell-killing membrane attacking complex called MACs is formed, and additional phagocytes are attracted. I'll show you the picture here. Okay, so this slide shows the entire process. See, the complement system can be activated by binding directly to the surface of an invading bacterial cell, and then starting this activated cascade reaction. Okay, this pathway leads to the form of this membrane attacking complex called a MAX, and the destruction of the invading cell. So this slide shows the MAX formed by the complement system. The MAX inserted themselves into the plasma membrane of the invading cell, forming these pores. So basically like a lot of holes, okay? So this causes water to flow into the cell by osmosis, and then eventually busting the cell and kill them. We call the lyse, okay? Okay, again, this is the same slide to remind you that now we start to talk about the adaptive immunity. It's a much more complicated process. As we mentioned, the adaptive immune response is the highest level and the last line of defense against the pathogens, okay? This system uses chemical and cellular reaction to neutralizing and destroy a pathogen. One most important feature of the adaptive immune response is that it has this memory components, okay, allowing our immune system to quickly respond to the same pathogen if it experiences second times of exposure. Okay, so this feature is distinct, very different to the innate immunity, which does not have a memory. Again, the adaptive immune response has two interconnected systems. One is the antibody, or called the cell-mediated immunity, and the B cells are the main cell type involved in antibody-mediated immunity, and the helper T cells, or sometimes we call it a CD4 positive T cell, facilitate the activation of B cells. But in the cell-mediated immunity, the cytotoxic or the killer T cells, or sometimes also called the CD8 T cells, is the major cell type and directs against the cells infected by pathogens, cancer cells, and transplanted cells. You would have to remember all these different types of cells. Okay, B cells and T cells are both lymphocytes in our body. So we talk about that. They are, a lot of them are in our blood. The B cells are produced in the bone marrow. Maybe that's the reason why I call the B cells, and migrated into the spleen, and then other secondary lymphoid tissues where they mature and differentiate into immunocompetent B cells. So that means they can function in our immune system. The main function of B cells, or plasma cells, once activated, is to make antibodies. So always you have to remember antibodies made by B cells. And then when they activate, become a plasma cells, they secrete antibody. Specific antibodies bind to antigens on the pathogen cell surface and can mediate antibody-dependent killing of pathogen cells. So interestingly, each B cell produces only one type of antibody. So if you need a lot of antibody for targeting the pathogen, you will need a large amount of antibody, right? So this is achieved by clonal expansion of B cells. We'll talk about it in a minute, okay? So in contrast to B cell, so T cells is matured in thymus, although T cells also originated in the bone marrow, but it's matured in the thymus, okay? Mature cells circulate and could be found in blood, spleen, and lymph nodes. All T cells produce T cell receptors, also called the TCR, which is a protein complex located on the surface of T cells. TCRs bind to pathogen proteins on the surface of infected cells, and this interaction mediates cell signaling and can result in killing of targets. Okay, so this slide shows several common features in these two systems. B cells or T cells both recognize an antigen. Okay, B cells use B cell receptor, or BCR, and T cells use TCR, right? So then both B cell and T cells, they can go clonal expansion when activated, meaning the interaction between B or T cells with antigen triggers mitosis, and then they build up of a large population of cells derived from the activated cells. Clonal expansion basically is from one cells, they will grow and proliferate into many stem cells, okay, clonal expansion. Okay, so because all cells in the population are derived from a single ancestor, they are clones. Okay, so these activated B and T cells, they can both attack and destroy the pathogens. Some of the cells can form memory cells, which can quickly activate the adaptive immune response if the same pathogen infects us again. So before I go into details of the adaptive immune response, I would like to introduce the MHC molecule, okay? MHC stands for Major Histocapability Complex. So MHC is expressed from a set of genes on chromosome 6 that encodes recognition molecules that prevent the immune system from attacking body's own organs and the tissues, all right? So in our body, there are two types of MHC molecules, MHC1 or and then the MHC2, class 2 or class 1. MHC1 molecules are found on all nucleated cells. So basically all the cells except for blood, red blood cell. We know red blood cell does not have a nuclei. So it does not express class 1 MHC molecules, but all the other cells, every single one, they express MHC1, okay? And then MHC2 is different. These molecules are only found on professional antigen presenting cells. What does professional antigen presenting cells mean? That means dendritic cells, macrophages, and B cells. So those cells, they can engulf the microbe or pathogen, and then we initially talk about it, right? So they will digest the pathogen and then make them into pieces, small peptide, and then this MHC2 molecule is going to present this small peptide on the surface, okay? So remember that when we talk about the neoantigen, right, we mentioned that the digested neoantigen peptide has to be presented to the cell surface for T cells to recognize, right? So the MHC molecule is the carrier of the short peptide and help to display the peptide to cell surface. So why they need to do that? So we're going to watch a short video to help you understand these two groups of MHC molecules.
A key feature of the immune system is the ability to distinguish self from non-self or foreign. This remarkable ability is necessary because our bodies are constantly exposed to pathogens, for example, viruses. The basis of its ability to distinguish between both is the presentation of antigens to immune cells. Once an immune cell recognizes its specific antigen, the body launches an immune response to protect itself. Antigens are presented on the cell surface by two protein classes, MHC class 1 and MHC class 2. Let's get started with MHC class 1 molecules. Cytosolic proteins are transported to the proteasome. A proteasome is a complex that degrades proteins into small peptides of approximately 15 amino acids in length. These peptides are delivered into the endoplasmic reticulum by a TAP transporter. In the endoplasmic reticulum, TAP interacts with the MHC class 1 molecule, which is linked to several helper proteins. The term MHC stands for Major Histocompatibility Complex and refers to the molecule's importance in determining compatibility in organ transplantation. When the MHC class 1 molecule permanently binds to one of the peptide fragments, the TAP transporter and helper proteins dissociate. The antigen-loaded MHC class 1 molecule is pinched off into a vesicle and migrates through the Golgi apparatus to the cell surface. Fusion of the vesicle membrane with the plasma membrane results in the release of the antigen MHC class 1 complex. The complex can then be recognized by effector CD8 T cells. Now let's take a detailed look at the MHC class 2 antigen presentation pathway. Exogenous antigens are taken up by cells with endocytic activity. In the endosome, antigens interact with MHC class 2 molecules. The binding site of these MHC class 2 molecules is initially blocked by a small molecule. However, another helper protein present in the endosome is capable of facilitating the exchange of this small molecule with any available antigen. The vesicle then travels to the cell surface, fuses with the plasma membrane, and presents the antigen MHC class 2 complex on the cell surface. The antigen can now be recognized by CD4 T helper cells. So what's the main purpose of these two antigen presentation pathways? Antigens presented by MHC class I are usually derived from inside the cell and are often referred to as endogenous antigens. These antigens can be derived from the cell, representing a normal self-antigen or neoantigens, which are derived from cancer cells. Other endogenous antigens include viral components from virus-infected cells. In contrast, MHC class II antigens are acquired from outside the cell and are therefore referred to as exogenous antigens, for example, extracellular bacteria.
Now I'll show you the process of the antibody-mediated immune response. In the stage of innate immunity, the phagocytes engulf the pathogen, which is the bacterium on this slide. After digestion, the MHC2 molecule presents the bacterial peptide, also known as an antigen, okay, to the cell surface, and then recognized by the TCR, the T cell receptor on this CD4 positive T cells. And this interaction stimulates the cytokine production from both the phagocyte as well as the CD4 positive T cells. And this interaction also further induces the clonal expansion of CD4 positive T cells, and then making a lot of those the same T cell clones. Since CD4 T cells are helper cells, this is how they help B cell activation, okay? Because B cells can also engulf the pathogen through BCR binding, so B cell receptor binding. So the MHC2 molecule in B cells will present the same peptide on the surface of the B cells. The TCR of the expanded helper T cell previously, we know that. So these helper T cells will recognize the specific antigen presenting on the surface of B cells, and then will link the two cells together, okay? Similarly, the interaction will stimulate the release of cytokines from T cells and activate the B cells proliferation, and then produce a clone of cells. Now this is a clone of a B cells, okay? Some of those clone B cells will differentiate into plasma cells, so which produce antibodies specific for this antigen, and a few differentiate into memory B cells. This slide summarizes the events happen in T cell activation and B cell activation. So this showing also on your textbook. So I will not get through those processes anymore. We'll just talk about it in those slides. This is a picture of B cell I mentioned earlier. Do you still remember those? So a mature but unactivated B cell, this is not producing any antibody. However, in these cells, this is a plasma cell. That means an activated B cell. So now it's very actively produced antibody. So you can see that in this unactivated B cell, the cytoplasm, it does not have many ER, endoplasmic reticulum. However, in this plasma cell is filled with rough endoplasmic reticulum. So this is associated with protein synthesis. Some B cells can produce up to 2,000 antibody molecules per second. So it's very highly efficient production of antibodies. Antibodies secreted by plasma cells are Y-shaped proteins that can bind to specific antigens. Okay, antibody has 4 polypeptide chains, 2 identical heavy chains shown in green, and 2 identical light chains showing in this blue color. So light chains and heavy chains, they are connected by this disulfide bonds, okay, showing here and here, Okay, so the molecule is Y-shaped and forms a specific antigen binding site at the ends in this place called a variable region. Sometimes they also call the FAB region. Okay, so the amino acid sequences of variable regions differ from one antibody to another, and that is where the specificity comes from. So in the exam, if I ask you where the specificity of antibody comes from, so you should know the answer is from the variable region, right? And then the constant region showing in this pink color exhibits a limited variation that defines the 2 light chain subtypes and the 5 heavy chain subclasses shown or shown in the next slide. So the 5 subclasses of immunoglobulins are called IgD, IgM, IgG, IgA, and IgE. So they are also named as isotypes, are classified according to the type of heavy chain constant region. Okay, and they are distributed and function differently in the body. Please remember that IgG represents about 75% of serum antibodies in humans. So it is the most abundant immunoglobulin in the blood circulation, IgG, okay? It involves in fighting with many kinds of pathogens such as viruses, bacteria, and fungi. Then IgE is the antibody or allergy and has anti-parasitic activity. Now let's talk about the cell-mediated immune response. In this process, CD8 positive T cell, also named as cytotoxic T cells, is the main cell type and it goes to clonal expansion upon activation. CD8 positive T cell can kill the infected cells directly by releasing enzyme perforin. Okay, this enzyme called the perforin. So perforin is inserted into the membrane of the infected cell, forming pores and then lyse the cells. Okay, why CD8 positive cell called a CD8? Because on the surface there has a expresses molecule, this protein called a CD8. All right. Let's take a look at the whole process. The picture on the textbook has some mistakes, so I used an online source to show this better. Okay. APC is antigen presenting cells we'll talk about, like a macrophage or the dendritic cells. Those APC cells, they engulf and present the pathogen peptides the same as we previously discussed, okay? So you have this antigen MHC complex. Interestingly, APC not only has MHC2 as professional antigen presenting cells, but also contain MHC1 that existing in all nucleated cells. These MHC1 molecules can also present antigens from the pathogen, okay? So the MHC1 and antigen complex can only activate the CD8 T cells, but not the CD4 positive T cells. Similarly, the MHC2 and antigen complex can only activate CD4 positive T cells, but not CD8 positive T cells. There's a trick to remember this, okay? So let's see, MHC1 pair with CD8 positive T cell. So you can think about the total number is 1 times 8 equals 8. And then MHC2 pair with the CD4 T cell. And then you can see the 2 multiply 4 equals 8 as well. So this is easier for you just to remember which one is interacted with which one, okay? But it is still not completely understood how CD8 T cells are activated. You may see different explanations from different sources. The latest studies showed that CD4 helper T cells are also involved in this process, okay?
The interaction between the CD4 T cells and APC strengthens the interaction between APC and CD8 T cells, which further activates and promotes clonal expansion of CD8 T cells. All right. So the activated CD8 T cells will find infected body cells. And remember, all nucleated cells have MHC1 molecules. Once they are infected, the pathogen peptides will be presented to the surface and recognized by these activated CD8 positive T cells. All right. So consequently, the CD8 T cells secrete the perforins and the granzymes, and then kill the infected the body cells. CD8 T cells can also target the cancer cells. As we mentioned, that cancer cells producing what? Neoantigens on the surface, right? So these are mutated proteins that are different from normal proteins made in our body, okay? Although it is not completely understood how cancer-specific CD8 positive T cells are activated, it's now generally accepted that when cancer cells die and are cleared up by the APC, the neoantigens can be presented by APC, okay? Consequently, the CD8 positive T cells can be activated and target these cancer cells that contain these neoantigens. In this picture, the T cells are showing in yellow and the cancer cells shown in red. You can see those CD8 T cells are attacking the cancer cell. We mentioned that an important feature about adaptive immunity is what? Memory. Always remember, it's important, okay? There's always a small fraction of memory B cells or memory T cells generated after clonal expansion, okay? So this is particularly important for vaccine. So this graph shows that antibody levels are increased after the first exposed to the one infection, but has a much faster and stronger response after the second exposure to the same antigen, okay? Conferring immunity to a second infection. So this is contributed by the quick activation of these memory cells, all right?
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
Now I would like to spend some time introducing what we know about organ transplantation, which is an important topic in immunology. You probably all know that a successful organ transplantation, you will need a matching organ, right? So this match means match of HLA haplotypes. Mismatched HLA haplotypes can lead to immune injection and transplantation failure. So the best chance for a match is between related individuals, and identical twins are the best match as they have the same set of DNA, right? So this picture shows the first successful organ transplantation in the United States in 1954, when a kidney was transplanted between identical twins. The surgeon named Dr. Joseph Murray was awarded a Nobel Prize for this medical breakthrough. The organ receiver, his name is Richard Henrik, showing on the right, okay, survived 80 years until the transplanted kidney has an infection and unfortunately died. Then what is HLA? Okay, HLA is human leukocyte antigen. So we talk a lot about MHC, and HLA basically is the same name for MHC, but it's in humans, okay? There are a set of 9 genes on chromosome 6. The picture shows 2 main classes of MHC molecules. One is MHC class 1. We learned this HLA-A, HLA-B, and HLC. And MHC class 2, that is HLA-DP, DR, and DQ, okay? Each gene has many different alleles, allowing countless allele combinations or haplotypes. We learned that a haplotype is a group of alleles that are inherited together. While we can only have 2 copies of allele or haplotypes per person, then there is very little chance that the haplotypes are exactly the same between 2 unrelated people. Indeed, among unrelated donors and recipients, the chance for a successful match are only 1 in 100,000 to 200,000. For the last section, I would like to introduce the disorders of the immune system. There could be an overreaction of the immune system, such as the most common condition as allergies, to a less common condition, anaphylaxis, which is a severe, potentially life-threatening allergic reaction. Other immune-related diseases are autoimmune diseases such as type 1 diabetes and lupus. This table lists a group of autoimmune diseases. Most autoimmune diseases are not curable. With more and more understanding of our immune system, we hope to provide more treating options for these diseases. This slide shows how allergy is triggered by an allergen. Showing here is the ragweed pollen, okay? So ragweed pollen is notorious for causing allergic reaction in humans, specifically allergic rhinitis. Up to half of all cases of pollen-related allergic rhinitis in North America are caused by ragweed. When ragweed pollen interacts with B cells, the B cells can make a large amount of IgE. Okay, IgE attaches to the master cells, and when the person is exposed to the same allergen, and then they regulate the pollen again. For example, the master cells produce and releases histamine, okay, which causes the characteristic symptoms of allergy. Many allergy medicines are antihistamine drugs, including like a Benadryl, Zyrtec, Claritin, and Allegra. Sounds familiar, right? By blocking the histamine signaling, these drugs can effectively reduce the allergy symptoms and will make you feel much better. So we talk about autoimmune diseases, right? So there's a rare severe type of immune deficiency called SCID, S-C-I-D. Also the severe combined immune deficiencies, okay? So these patients have defect on both antibody-mediated immunity and cell-mediated immunity, okay? Infants with a SCID appear healthy at birth, but they are highly susceptible to severe infections. One famous the SCID patient, his name is David Vetter, had been named as the boy in the bubble because he lived in an isolated this bubble-like equipment for 12 years. Unfortunately, he died of complications after a bone marrow transplantation from his sister. The autopsy revealed that his sister's bone marrow contained traces amount of a dominant virus. It's a human herpes virus, which had been undetectable in the pre-transplant screening. So this is a very common virus, as half of all 5-year-old children and about 90% of adults in the United States have evidence of a previous infection. Okay, but David's sister did not show any symptom. However, the virus was deadly for David. Okay, this is kind of a sad story. So David Vetter was a Houstonian, and now there is a clinic named after him at Woodlands. This is a David Clinic. Actually, also an elementary school which opened in 1990 in the Woodlands was named David Elementary after Vetter. Okay, maybe some of you may know that. Lastly, I would like to spend a few minutes briefly introducing AIDS, or this acquired immune deficiency syndrome. Okay, so this is an immune-related disorder that is caused by HIV infection, the virus HIV. So this disease is especially hard to treat because the HIV virus infects and destroys the CD4, the helper T cells. We already learned that today. We know that the CD4 T cells are essential for our adaptive immunity, right? So once the disease goes to an advanced stage, the numbers of CD4 positive T cells significantly decrease, okay? So then the patients can be easily infected by viruses, bacteria, or other pathogens. So the weakened immune system can result in death for the patients. HIV is a RNA virus, okay? So meaning the core of the virus are composed of RNA, this is viral RNA shown here. The viruses contain this lipid envelope with glycoproteins named P120, which can interact with CD4 and the CCR5 molecule on the surface of CD4 positive T cells and can get into CD4 T cells. The virus can make reverse transcriptase and therefore convert its viral RNA into viral DNA. Okay, it used to be RNA, but then can make a DNA. And then this DNA will integrate into the host genome, like a human genome, okay? So this is particularly dangerous when the genome is inserted by the viral DNA, because if the T cells are infected but not lysed or destroyed, these viral DNA can serve as a latent viral reservoir and started to make new viruses whenever condition is permitted, okay? That is the reason why HIV-infected patients can hardly be cured. The current cocktail therapy can inhibit the virus from being expressed, but cannot completely eradicate the latent virus. They have to take the medicine for the rest of their lives, those patients, okay? So scientists are working hard trying to find a solution. So if I ask you, does HIV change the genetic information of its infected cells? You should know that the answer is yes, right? Because they can do this because they inserted into the host genome, right? But then I'm asking you, can this altered genetic information be passed to next generation or the offspring? The answer should be no, right? Why? Because HIV only infects CD4 positive T cells, and the human germ cells do not express the CD4 molecules. Therefore, they are not going to be infected by the HIV, right? However, you probably heard that the babies could be infected with the virus. Why and how? That could happen if there's a direct blood contact or breastfeeding, but not through the inheritance we learned from this class, right? Okay, so a side note is that the COVID-19 is also RNA virus, right? But it does not integrate into our genome. So this is very different from HIV. Therefore, theoretically, it should not come back after being cured. So theoretically, COVID is similar to flu viruses or common cold viruses, right? HIV or the AIDS is also a pandemic, okay? Currently, approximately 38 million people are infected worldwide. Our textbook was published in 2015, so its table shows that 35 million people were infected. But in some of the African countries, such as Swaziland and Botswana, 1 out of 4 to 5 people are infected with HIV. Apparently, we have to work hard to find a solution for this disease. All right, that's all for today.